want to welcome you all back to continue the morning of wonderful talks. I'm Paul Blaney, faculty member here at MIT, member of the Koch Institute, and also at the Broad Institute. Before we get started, I have a housekeeping note to pass along from the organizers. As our seminar culture has an evolving courtesy around taking pictures of slides, the request is for everyone to make sure the sounds are off on their phones. So if you do choose to capture some images, it doesn't make a noise that disturbs others. And of course, if a slide says, do not post, we ask that you do not post. So we'll turn now to introduce our next speaker, Dr. Sydney Shaffer. And Dr. Shaffer fits in really well in this community, being trained as a chemical engineer, but working in biology of interest to this group. Dr. Shaffer is an assistant professor of pathology and bioengineering at UPenn, early independence awardee from the NIH, and established her group at Penn back 2019. We were chatting at dinner yesterday a little bit about that pandemic lab startup experience, which I can only imagine. She works in an interdisciplinary fashion across a range of areas, including single-cell genomics, lineage tracing, using bioinformatic techniques, applying all this to cancer biology, studying cellular heterogeneity, both in normal tissues and under disease conditions. And so I will pass it over to Dr. Shaffer for her talk on tracking cell state plasticity in injury and oncogenesis.
Great. Thank you, Paul, for the lovely introduction. And of course, thank you to the organizers and Matt for the opportunity to speak here. Really very honored. Today I was going to talk about a story from our lab that's more recent about thinking about cell state and how it changes in time and what happens in the setting of injury and at the very beginning of cancer. I like to start with this example of the butterfly. So I did this activity with my daughter where you order caterpillars and you grow them up in your house. And what you can do is watch the stages of the life cycle of the butterfly. And for a child, this is a really cool activity because you get to see how biology changes through time. But in reflection on the act of doing this with her, I started thinking about this a little bit more because this is, analogous to a lot of the things that we do in biology. We look at pieces of information, we get individual snapshots of processes, but these processes are dynamic in nature. And so when we take snapshots, we get one piece of the puzzle, but there's a lot of value in really understanding how things evolve and change through time. And so, with this activity, you can do direct observation. It's a lot like doing like a microscopy experiment, but, different scale. And you can watch things change. And so today I'm going to make the argument that when we do a lot of our single cell assays, we really are missing this element. And we just get the one snapshot. And what we really need is a better understanding of the relationships. And of course, there are many ways that we can do this, both experimentally and computationally. And so this problem is compounded by the extent, and this is the topic for the symposium today, of heterogeneity that exists in cancer. And, as we've gone from, one gene at a time kind of measurements to many genes at a time measurements, we can see that cancer cells are very heterogeneous in nature. And what would be really meaningful, or what is, really helpful, is to understand how this heterogeneity arises, where it comes from, and the relationship between heterogeneous populations that we see. And so this has been a huge interest of my group. We want to take single cell data and we want to find what is the function of these different cells. What are their, what are they going to do? And so we like to ask the question of what do different cells do in our data? And the way that we've approached this generally is through the use of techniques that we call lineage tracing. And so lineage tracing is the way that we can, take the butterfly and understand the stages that came before and the pieces of that, And there's many approaches to this. And so there's the direct observation strategies, there's reporter systems like we've seen. There's techniques where you put in barcodes or labels, pieces of DNA that you can use to track through time. There's evolving methods that change through time and provide even more resolution. But where I think this is really exciting is also when we can start to then take lineage tracing methods and pair them with genomic assays. So not only do we build that lineage tree, but we can also say, okay, we have the lineage information, and what does this look like at the cell state level or at the chromatin level or accessibility or whatever measurement that we're interested in. And so we're really interested in integrating these with genomics. And so how do we think about this? When we start to build these trees, one of the things that we can do is we can find things that show memory. So we can find related groups of cells that are actually transcriptionally quite similar to each other, and we term this memory. We can also find things that change through time, and this could be intrinsically changing or driven by an intrinsic cue or driven by an extrinsic cue, but this we can set this up in contrast to things that show plasticity. And here I refer to plasticity both as, in response to perturbation, but also that could be in the absence of perturbation, but naturally arising or just arising organically. And so a lot of my group has worked on thinking about memory and plasticity and using lineage tracing tools to do this. And we've done previous work that I'll mention briefly, looking at this in the context of drug resistance. We've also thought a lot about state switching and how that leads to resistance. But a lot of this, my prior work or prior work of our group has really been focused on doing this with like barcoding or synthetic tools. But the problem with synthetic tools is we can't actually use them in human systems. And so that's where this story or this project really started, was to start to look and think about how we can take this framework of studying plasticity and memory and apply it in the context of human tissues. And so we became interested after this really nice paper from Leif Ludwig in 2019 in the use of mitochondrial mutations as a synthetic barcode that could be used to track cells from human tissues. And so there's a lot of things that are really favorable about sequencing mitochondria. You have a relatively small template. So it's a small piece of DNA to sequence, or RNA, or however you're getting it. It's a small piece to sequence. There's also many copies of DNA, and they undergo somatic mutations. And I like to highlight here that with the data I'm going to show you today, we're talking, these mutations occur at varying allele frequencies. And so this is because you have many copies, and so this is referred to as the allelic heteroplasmy. People have, of course, used mitochondrial mutations before to look at tissues, but the challenge with them was that these were often done as bulk measurements, and you would have mixtures of cells, which would make it hard to know which cell had the mutation, or you wouldn't know how to interpret the allele frequency itself. So you could have a mutation that's really highly present in one cell or a subset of cells, or it could be present at a lower level across many, many cells. And so distinguishing those possibilities requires single cell resolution. And so I also just like to pause and talk about, I told you the strengths. What are some of the limitations of using these techniques? You don't know when the mutation happened, so you can't actually pinpoint that. People have done really nice studies showing that a lot of these happen as people age, especially in stem cell compartments in tissue. So the majority of the mutations that we're capturing, we presume to be age dependent and not arising during development. Different cell types have different number of mutations, so you have to take that, different numbers of mitochondria, so you have different loads of the template. You have to worry about coverage and capture and how much of it you can pick up with your technique. And then there's also selection and drift that can happen. You can have selection for a mutation if it's favorable for that mitochondria itself, and so on. I will stop and say that I think this is an opportunity for orthogonal methods, measurements as well, and my group has been working on that. Unfortunately, I don't have any of that to show you yet today, but there's other ways that we can integrate this with other lineage tracing tools and provide even higher resolution. So what did we do with this? We started by looking at metaplasia, as it is an example of plasticity. Metaplasia is when you have one mature tissue type that is replaced by a completely different mature tissue type. And so here I highlight metaplasia of the esophagus, where we have stratified squamous tissue that gets replaced by a glandular tissue that looks a lot like the intestine. And those were my simplified diagrams. Here's an example of a H&E from these tissues as well, to give you some context. You can see the normal stratified squamous gets replaced by this intestinal-like tissue. And we picked this for a number of reasons, but one that actually ended up being very much to our favor, working out in our favor, is that actually the esophagus has a really high number of specific mutations that occur in it, representing potentially somatic clones. And so this tissue has actually also, measurements show it has a high number of mitochondria, but it also acquires a high number of mitochondrial mutations. And so why do we care about metaplasia? Well, metaplasia is a response to injury. Patients who develop metaplasia generally have reflux disease. They're having acid and bile reflux in their esophagus, but it also predisposes these patients to cancer. And so these patients are at higher risk of progressing to cancer, and they get regular monitoring. This is actually really helpful for a number of reasons, one of which is we can get access to this tissue fresh from people before they have developed cancer, and we can start to look at lesions in their earlier stages. And so that's another practical consideration for Barrett's. And so Barrett's sits as part of this continuum. You have the normal squamous epithelium of the esophagus on one side. Patients can develop Barrett's. You get this intestinal metaplasia. Many patients just stop at Barrett's and have stable Barrett's for many years, but a subset of patients will develop dysplasia and eventually invasive cancer. And I will say the dysplasia has all the morphological features of early cancer lesions. And so we designed this study to begin to look at the transcriptional states and the mitochondrial mutations in these patients. And so we collected endoscopy samples from 13 patients. We collected from the adjacent normal esophagus, the adjacent top of the stomach, and from the Barrett's lesion itself. We worked with two phenomenal collaborators, Gary Falk at Penn, who's an endoscopist, and Amanda Meyer, who is a pediatric gastroenterologist, but also runs a lab that develops some organoids. And so from these patients, we took them fresh biopsies back to our lab. We disassociated them. We did single-cell RNA-seq, and we also prepared the mitochondrial enrichment libraries. We used actually this technique called MASTER, where we use the standard 10x workflow, but what we do is we do a targeted amplification of the mitochondrial cDNA out of the mitochondrial cDNA out of the main cDNA library. And this allows us to get separate, really high coverage of the mitochondria that's independent from the regular RNA. So we started with looking at normal tissue in the transition to Barrett's. So a big question in the field is, what is the cell of origin of Barrett's? And so there's been a lot of focus on this idea that there's one cell of origin and that it gives rise to Barrett's esophagus. And people have proposed different stem cell populations that could be representative, or they could be potential progenitors for Barrett's esophagus. And so when we started to look in our data, one of the first things that jumped out at us is, across many, many patients, these tissues are really different from each other. This is all of our patients actually put together. You can see the pink area is our Barrett's esophagus, and it's separate from our regular esophagus and separate from our stomach. We have supporting populations as well. But when we look at, when we look at the mitochondrial mutation data, we break this down and we can look at this on a patient-by-patient basis. So here we have a single patient from this cohort, and each of the different rows is representing a different mutation. The allele frequency is measured in the blue, and we can see somatic subclones that have arisen in each of our tissues. And so the orange corresponds to the esophagus, the pink corresponds to the Barrett's, and the purple corresponds to the cardia. And each one is separate from each other in these measurements so far. We can see this across many patients in our cohort. But actually, one of the things that was quite interesting is when we zoomed in more closely to some of these mutations, we actually pick up mutations that we can capture across different tissues. And so here we worked with Nancy Zhang's lab at Wharton, and we developed a model to say what's a variant that we would call a distinct somatic subclone to discriminate it from a modeled background or predicted background. And when we apply this, what we can see is that we have clones that are present in our Barrett's esophagus lesion that overlap with the neighboring tissues. And I show one patient for demonstration first. I will also note that we don't see this clone present in every single cell from that patient. It's really just a subset of the cells that we're capturing the clone in. But we actually see this phenomenon generally across many patients in our cohort. And it's not just Barrett's esophagus clones that are shared with their neighboring tissues. It's actually also clones that cross the esophagus and gastric junction. So we have mutations that we're picking up in these two distinct mature tissue types. And I will say there's many of these. It's not like, we're trying to analyze 5 cells from this one and 5 cells from that one. In this one patient, we have many separate mutations. In other patients, we have bigger mutations that have expanded. We're also seeing evidence for this in other orthogonal measurements that I was alluding to before. We can also see this in other sequencing data as well. And we capture these across all 3 of these different tissue types. And so I bring this back up to highlight that we really don't think that this single cell of origin is the right paradigm here, and that it's actually more complex, and there's a lot of plasticity. And we have been able to capture clones that suggest the esophageal and gastric origins can contribute. We also did something in one patient in particular where we looked at a bunch of biopsies that we took at discrete measurements all the way down the esophagus and into the cardia, and made measurements along the way. And one of the things that we found was that there was a variant in this patient that we picked up at many different positions along the gastroesophageal junction. And so you can see this variant is highest when we approach the gastroesophageal junction, suggesting that that's the highest density of it, and that then these cells have really spread and migrated and contributed to both the neighboring tissues there. So this patient in particular further confirms that we can connect the clones across the esophagus and cardia. We also picked up this interesting population that sits between that in our UMAP space sits distinct from both the esophagus and the stomach, but it actually, we think, represents a transitional junction in the tissue. And so we find in this population, they express markers of both the esophagus and the stomach, but they also express some unique markers. And it actually looks a lot like what has been described at the squamocolumnar junction in a mouse model as well. And so we think we've picked this up in human tissues. And when we look at the mutations in these cells, we can find that many of the cells that sit at this junction position, indeed, they have a mutation that we can then pick up contributing to both the neighboring esophagus and gastric tissues. And I show a separate sample from the same patient just to show that we don't pick it up everywhere. It's just in the biopsies that we took that were near the junction. And so we find that cells of the esophagus, gastroesophageal junction, and stomach can share the same mutations. And I bring this up to just highlight that this might be a feature of epithelial boundaries where you sit. If you're near that boundary, you can the location to that boundary actually matters in determining cell types you can become. And one thing that I want to highlight about this data is that this is all taken from patients that have reflux. And so presumably these patients have had a lot of injury and regeneration through time. And many of these were tracked. They've had many, many years of reflux. And one of the ones with the most extensive mutations also was not on PPIs, which is kind of interesting. So if we look more at Barrett's metaplasia, we find that Barrett's itself has many mature cell types in it. It looks a lot like these intestinal cell types, and is these cell types that are normally present in the intestine. We can find specific markers of them, but when we look across patients, we find that most patients make all the different cell types that are present in Barrett's esophagus. The exception I will come to in a minute being patients with dysplasia. So patients with diagnosed non-dysplastic, no dysplasia, they make all of the mature cell types of Barrett's. And so we asked whether all of these mature cell types can come from the same common progenitor. And to do this, I'm going to just show one patient, but we see this across many patients in our cohort. If we look at the cells that are present in there, we can find multiple mutations. And within these mutations, we can show that any one of these clones can generate all of the cell types. And these exist at similar frequencies to the overall composition of the Barrett's. And so from this, we can show that these cell types can come from a common progenitor. And so in these tissues, we find a single clone can generate multiple cell types, but then also multiple clones are contributing to the metaplasia on a per patient basis. So one of the things that we've been doing is, we've been looking at all of this in disassociated tissue. And so we were really interested in what does this actually look like in the native tissue context. And so one of my students recently set up this protocol to work with SlideSeq and to perform SlideSeq and spatially sequence the RNA present in Barrett's esophagus, but also to capture the mitochondrial mutations. And so what she was able to do was, in this patient, sequence, look at the structure of the Barrett's esophagus itself and then say, what does one of these clones really mean? And so what we think we're capturing, we are capturing this mutation. We find that it's in all the cells that have the mutation are in close spatial proximity to each other. And we think this corresponds to a single gland in the tissue. And when we look on the H&E, which is a little bit distorted, this does look more like a single gland. And we don't just pick up this one, but we pick up multiple of these different little pockets of mutations where presumably there is a progenitor cell that harbors that mutation. It differentiates and makes all of the progeny that have the same mutation in them. And so when we are picking these up in our tissues, we think these represent little pockets of mutant progenitors in the tissue. And we did DNA sequencing on a JSON slide to confirm this. And so what happens when they go on to dysplasia or the beginning of cancer? And so here was actually a patient that I like to highlight because it was a nice example of one where we got actually the non-dysplastic tissue and we got the dysplastic tissue in the same biopsy. And when we look at these patients, we can really ask, how does the dysplasia itself evolve? So does the whole thing convert or part of it convert, or do we have now clonal sweeps that we didn't see at the origins of Barrett's? And this is where the mutational tracking comes in. And so when we looked in this patient, we could then start to see the beginning of clonal sweeps. So here on the left cluster, we have the non-dysplastic cells, and you can see there's very low allele frequency of this mutation actually up here in this compartment that expresses some of our stem cell markers. But then in all of the cells that are part of the dysplasia, we find very high allele frequency of the mutation, pointing to a model in which we have a single progenitor that gave rise to all of those cells that are part of the early cancer lesion. And we find other subclones as well that are coming out in this tissue and have distinct transcriptional states that are different from the initiating clone. And so here we also have a subclone within it that has emerged that we can see. And then there's actually a third subclone in this data as well that has come about. And so we can start to see the clonal evolution. And in other patients, we can also capture these clonal sweeps as they happen in the tissue as well. And so in this case, we wanted to ask what are the transcriptional differences leveraging our single-cell RNA-seq data. And one of the things that we found is that these dysplastic cells actually have really high expression of LGR5 and NOTAM, pointing to Wnt dysregulation as a potential target for the dysplasia. And so we went back in and looked at this in situ, and we can indeed find high expression of these Wnt targets. Given this, both pieces of information, that we have this founder clone and that we have these Wnt changes, we asked whether there was a possible Wnt mutation. So on a number of these samples, we also performed whole exome sequencing from the same biopsy. And in this patient, indeed, we actually found a mutation present in APC that is presumably driving the Wnt dysregulation. And I'll breeze through this in the interest of time. We also, in our other subclone, we can also find associated transcriptional changes that are enriched in that subclone. And these also relate to potential mechanisms of increasing fitness in these clones. And so finally, given this transcriptional state information, we then asked whether we could see evidence of these states present in late-stage esophageal cancer. And so for this analysis, we pulled data from TCGA. This is bulk data, but we ran a deconvolution tool called cellular signals analysis that actually allows for an intercept term, which is a really nice thing because a lot of deconvolution tools don't have these. So when we run the same analysis on breast cancer data, everything basically goes to this intercept term, which is a nice control that this is actually something specific to this cancer type. And you can see, if you look at this, the strongest signal is cycling, which makes sense. Like our cycling cell types in our data set look most like the cancer. But we also have this subset over here that show enrichment. It's just a subset of all the samples in TCGA, but they show enrichment for a signature that has our Wnt pathway genes, LGR5 and NOTAM, as well as others. And so we think that this represents a subset of the ways that dysplasia can evolve to esophageal adenocarcinoma. So wrapping this up, with lineage tracing and single-cell RNA-seq, we can build a more complex picture of the evolution of Barrett's. We found that normal tissues and Barrett's can share the same mutations. Barrett's itself consists of many clones, similar to their adjacent tissues. We find that non-dysplastic Barrett's contains these well-differentiated cell types that come from one progenitor, and that dysplasia can come through a single clone that expands, and this represents a gain of fitness, and we can find additional subclones that can emerge as well. And with that, I would like to acknowledge our wonderful collaborators, Amanda Meyer and Gary Falk and Nancy Shang at Penn and Chop. And I'll take questions. And this project was led by Rodrigo Guiar, who was a previous lab PhD student.
Great questions. We'll start right here.
Great tool. One the fact that was kind of interesting is whether you are actually seeing transfer of entire mitochondria around your initial lesion, because you see that in cardiomyocytes, and you also see that in osteoblasts, where the entire mitochondria is transferred from one cell to another.
Yeah. So in general, are you okay? Yeah. In general, we can't rule out that mitochondrial transfer doesn't contribute to some amount of the variation that we see. In general, though, the loads that we're seeing of the mutations that we're using for the conclusions in our analysis are really high allele frequency loads. So we're looking at when presumably that mutation has fixed almost all of the mitochondria present in that cell. So actually, that's one of the complications with looking at lower allele frequency variants that just is really hard to account for. And I think we do see other signal in our data that is much lower, that is probably representing mitochondrial transfer, but that doesn't surpass the cutoffs of our model.
Great. And we'll alternate sides here.
So it was a very interesting talk. So did you had, by any chance, a temporal sampling of the tissue during the metaplasia? Because I was wondering whether when you transform the squamous epithelia into a glandular, it disturbs the barrier. And then you allow some glandular cell to intermix with the glandular epithelia that come from the metaplasia. And so what is the time of moments in this process?
The time of what? Pardon?
The time of the progression, Did you find, for example, in very early lesion, some cells that share a common ancestor with the glandular epithelia, or is the glandular epithelia come in the later during the process, when you allow this intermixing in set of similar histology? Yeah.
So it's really hard to know if it's time. We do see what you're describing. We definitely see these clones that we can capture in both the squamous and in our glandular, and we can see them in squamous that is starting to look like it's dedifferentiating. It's hard to know how long anybody's had Barrett's. So associating it with time is a real challenge here. We have started to look at squamous islands and features like that as one way to get at this, where, but again, we're still in the same conundrum as we don't know how long the patient has really had it. So I feel like that's the challenge there. But yeah, no, it's a good point.
Take two more if they can be quick.
Sure. I was very intrigued by your statement on plasticity, that you think potentially multiple clones contribute to the metaplasia. And I wanted to make sure I caught that correctly, and I was wondering how you picture that working.
Yeah, so we can pick up independent clones in the same patient that are from the esophagus that give rise to the metaplasia, and from the stomach that give rise to the metaplasia. So I think that tells us that we don't have one single progenitor that swept everything, which is something we never pick up in a patient that we've sampled with non-dysplastic Barrett's. You could say we don't have the mutation, that's totally fair, but the fact that we have independent clones contributing. So I think if you're locally in that region, sitting in that milieu around those neighboring cells, you have a higher potency potential to transition. I don't know if every cell can equally do that. Maybe it's just the stem cells when they get close to that milieu that are capable of that. I think these are all questions we have to ask. But I think this reflects really pretty profound field effects that are going on in these tissues. There's also some really interesting observations when people do ablation and repair that would suggest this as well.
Wonderful. Thank you.
Thank you.
All right, one more.
Can you comment on the functionality of your mitochondrial mutations? Because it looks like heteroplasmy is going down, which might argue selection for some of these. And so are any of them functional based on where they're located in the mitochondrial genome?
So we've taken a lot of these, or we've taken the majority of our cohort and looked at their predicted function in the population, or in a database, sorry. And there's the one that actually expands pretty broadly that we see across many of these is thought to be a fitness gain. And so we definitely are picking up some that have fitness gains. Whether they're being selected against, I think, is a harder argument to say. Nothing comes out as a home run in these databases that's being selected against. I can say that none of them get picked up across multiple patients, so we're not picking up anything that's recurrent, always driving, always leading to some function every time they're independent of each other thus far in our cohort.
All right, let's thank Dr. Shaffer one more time.
Oh, you have your, okay.
All right, and it's my pleasure to introduce Dr. Alex Shalek for our next talk. I am looking at a document that says Shalek Short Bio. It's not that short. It contains words I'm not sure I can pronounce. But I'll do my best to run through this and not take too much of his time.
Why don't you just say this is Alex?
Because this part's fun. Dr. Shalek is the J.W. Kieckhefer Professor in IMES. I'm going to shorten as much as I can. And the Department of Chemistry. Member of the Koch. Member of the Broad. Member of the Reagan. And an assistant in immunology at MGB, as well as an instructor in HST at HMS. All right, so I got through a lot of it. Alex studied in chemistry and chemical physics, so continuing our chemical theme for speakers in this session, and transitioned into genomic bioscience with Hong Koon Park and Aviv Regev at the Broad during his postdoc. Alex is going to give us a talk this morning titled, the title changed, Characterizing Cell Intrinsic and Microenvironmental Drivers of Cell State, Plasticity, and Drug Response in Cancer. This is Alex.
Hey. Thanks, Paul. I try to make that as short as possible for you. Thank you all for coming. I'd normally say thanks to the organizers. I'll just say thanks to Mike and Matt for actually allowing me to speak here, and thanks to everybody that came to present. It's really cool, and I'm going to continue on some of the themes that came up this morning. And what I really want to talk about today is the idea of the importance of cell state in cancer, talking about what drives it, what drives its plasticity, and how it is implicated in response. So like many, I have disclosures. I'll talk about funding on the last slide. The others aren't relevant to my talk today, but they're there for transparency. So before I begin, I thought I'd just introduce what our lab does broadly to this audience. What we're fundamentally interested in is the idea of balance in our tissue ecosystems. This has come up in some of the talks earlier today. If we were trying to figure out what constitutes balance at a cellular molecular level, we'd be really hard pressed to do it. But we know it's important because if we get too much immune activity, we can get tissue damage and death. And if we have too little, we can become susceptible to the outgrowth of opportunistic infections and tumors. So we study a lot of systems, trying to figure out what balance really means and what are the deviations that are induced by different environmental exposures, and then thinking about how we can understand how to restore balance as a cure or buffer in the first place as a prevention. And to do this, we spend a lot of time developing and applying tools that help us understand what's happening in these complex ecosystems. And as you've heard about from the others this morning, this has been an incredible time to think about cells within tissues. And that's because over the past 5 to 10 years, there have been these transformative advances in cellular molecular biology and in micro nanotech that now let us profile cells at unprecedented resolution. So we're no longer just thinking about the DNA of an individual cell or of a person. We can think about the DNA of an individual cell. We can also ask questions like what regions of that DNA are being read, what are the proteins that are expressed on the surface, what does the entire transcriptome look like? And we can actually measure multiple of these things in parallel. But the question becomes, when you can measure everything, should you? And really, in this morass of everything, what is it that really matters when we want to think about curing cancer? And so if we think about where we would traditionally come at this from the history of cancer, we would have said, well, DNA mutations are sort of the most important thing because we're thinking about something that separates self from non-self. And so, in many places, we think about what are the genetic mutations that exist in our DNA. We go and try and create models that recapitulate them. And then we try and figure out what are drugs we could use that would act on those mutations. How well have we done with this? Well, in some places we've done pretty well. There's some patients that have actual mutations, and we have really good examples of places like BCR-ABL where we can use drugs that go against these fusion genes. But the problem is that many of our cancer patients don't harbor actionable mutations. And in many places, we evolve resistance or we develop non-genetic mechanisms. And so I think, when I think about this, the funny thing is that when we think about the idea of targeting cancer, we're very often focusing on the idea of DNA. But at the end of the day, we want to actually kill a cell. And so when we think about this, we have all of these layers that take us from DNA to a cell. And that's why we can have the same DNA in each of the cells of our body, but get a variety of different cell types. And what makes this happen are many of the things that have come up this morning in conversations. So things like epigenetics, interactions with the microenvironment. And this results in different RNA programs that can lead to different cell states. So analogously, you could probably imagine in the context of cancer, that differences in epigenetics, interactions with the environment could lead to different cancer cell states that might have different phenotypes or different responses. And so we've become really interested in understanding what RNA states exist in tumors, what are the factors that influence them, and how do those couple into drug responses and patient outcomes. And so I want to tell 2 quick vignettes and then tell you about where we're going, focusing on some work that we did as part of a cancer systems biology consortium that was funded by the NCI with partners at the Koch Institute and the Dana-Farber, that really thought about trying to think about cell intrinsic and extrinsic mechanisms of drug resistance, bringing together a number of different technologies like cell mass and growth rate, some single cell genomics, some functional assays, multiple different models from organoids to PDX to primary samples, as well as some computational methods. And so I'll start with the cell extrinsic side because it's a little older, and then I'll go to the intrinsic, which we just put up on bioRxiv, and then tell you about what's going on now. So, one of the places where you would think of that a targeted therapy should work really well is in pancreatic ductal adenocarcinoma, or PDAC. That's because when you look at the genetics, it has these relatively uniform properties where 90% of patients have KRAS mutants, 75% have P53, and there are a number of other ones that show up. But this is a place where genetic targeted therapies hasn't worked very well. There's one drug that targets the G12C mutant of KRAS, and that's a blockbuster, but that's 1% of 90%. And so you might ask, well, if genetics doesn't help here, what do we look at? Well, most people have started to look at RNA, and what we find when we do this, if we look at tumors, which are columns here, is that some tumors express basal or stem-like genes, and some express classical or epithelial-like genes, and some are a little bit more ambiguous. Now, why is this important? It's because when we look at people that are either basal or classical in their phenotype, they have different outcomes. So more epithelial-like does better, not by much, but basal does worse and has greater resistance to chemotherapy. So you might ask, well, are these states actually going to be important, and do we preserve them in models? Could they help us think about, targetability in addition to prognosis? Well, we went to look at the models that existed, and what we found is that when we looked at primary tumors or metastatic tumors and compared to cell lines or organoids, that all of the mutations that we expected showed up in the distributions that we would have expected in our models. But at an RNA level, we saw that things were very different. We saw that in our patients that we would find some that had these classical-like phenotypes, some that were more basal-like, some that had some other signatures, which might be due to infiltrating immune cells. But our cell line models were all basal, and our organoid models were all classical with some other signatures coming in. And so what's super interesting about this is that it told us that neither of these systems were capturing our cell states. There probably was something interesting about how we were making models and how this came together. So you might be asking, why does the model look different? And we thought, well, maybe it's selection, maybe it's plasticity, maybe it's loss of interactions. And so we did an experiment to actually get at this, taking a small biopsy, using some of the technologies that you've seen earlier today in other talks, profiling those cells, making matched models, and looking to see what is preserved and what's not. I'm going to summarize this story really quickly and just get to some of the punchlines, given the amount of time I have. But in work that was led by Pete, Andrew, Hannah, and Shri, we took pinch biopsies from metastatic PDAC liver lesions around single-cell RNA sequencing and found that there was a spectrum of states, much like you see in a number of different cancers, where we had basal on one side, classical on the other, but also cells that co-express both programs. When we made the models and looked at them over time, we found that there were cell states that were missing that I'm showing you right here, so they were primarily classical. And by playing with factors in the media, we showed that we could actually recapitulate what we would see normally. So leveraging that plasticity to get back models of exactly what we would see ex vivo. Now, the thing that I want to highlight in looking at this is the functional impact of these states. And so not building this up really quickly, what I'll tell you is much like you saw this morning, by manipulating the microenvironment, we can manipulate the states while holding the genetics constant. So here's one particular organoid that we made where it started off with, in the classical state, you can see each point is an individual cell. And by adding one of the microenvironmental factors that we think is important, TGF-beta, what you see is over time, we drift to a state where we are primarily co-expressing the two programs and a little bit more basal. And when we strip away TGF-beta, we go back to where we were to start. Now, what's so important about this is that when we start screening frontline chemotherapies like gemcitabine, what we find is that in the classical state, we're highly sensitive. When we go over the basal state holding TGF-beta constant, we become much less sensitive, and when we remove it, we go back. And we see that this holds for a number of different drugs. But it's not just the basal state is more resistant. Actually, as we look across a large panel, taking isogenic models and toggling them back and forth, we find that the classical state's much more sensitive to chemotherapy, and the basal state is much more sensitive to MAP kinase inhibitors that hit MEK and ERK. And what's so funny to me about this is that we're seeing these major changes in helping us to select what would be an important drug, but these are things that we never model when we go out and do some of the functional screening, some of the testing that comes up that helps us to go from those observations that Sid was talking about to causation. And so, if I was trying to give you the no-doubt synopsis of this, the point would be that if instead of targeting mutations, you target the pathways that cells are using at a given point, you can do a better job of driving the outcome that you want. Now, you might say, well, does this help you select, which patients you should use these drugs on? Well, our data suggests that if we look at these things, that the classical individuals would be much more sensitive to chemotherapies. So you can go reanalyze existing clinical data, and this is exactly what you see. The tumors that regress are the ones that are more classical-like, and the ones that progress are more basal-like. We can also play this same game to figure out what are things in the microenvironment that might be driving plasticity. So we can look at factors that are secreted in a paracrine fashion that would be up in classical-like tumors, up in basal-like tumors, and those that show up in co-expressing. We find, for example, that gamma shows up in co-expressing. And so if we dump gamma on, which would normally come from T cells, you might say, well, will this move to being co-expressing? And the answer is yes. We can manipulate these things around. And you might say, well, is this something that happens in vivo? Well, it's hard to test this, but we had that one opportunity, getting back to something that Cedric brought up, where we were able to do a longitudinal biopsy. We looked at pre-treatment with an immunotherapy, and we found that most of the cells were expressing classical genes, but post-immunotherapy, we had shifted to co-expressing in basal genes. And so when we look at the differential expression, we see interferon is up, basal is up, classical is down, our T cells are secreting things that look like they're activating gamma. And so what's so funny is that by giving immunotherapy compassionately, because we think that this is going to work well here, we've actually driven, this cancer into a more aggressive basal-like state. But at the same time, we've made it more sensitive to MAP kinase inhibitors. So if only we knew to use that in combination with immunotherapy, as opposed to using chemo plus immunotherapy, we might get better outcomes. And at a GRC last week, I saw some data suggesting this actually was in fact the case. So I summarized this really quickly because it's a little older, but the idea is that microenvironment is really important and that we need to model it because it impacts drug responses. And this paper, I hope, provides a framework for thinking about how to model these things and think about their role. Getting back to some of the things that came up earlier. So I wanted to transition to talking about the idea of the environment to thinking about cell intrinsic mechanisms. And this is a paper that we just threw up on bioRxiv last week, which was asking sort of the opposite question. What if instead of going to a place where we know that there's common mutations but they don't work, what if we go to a place where there are mutations and we know they work? So what if we go to, for example, adult BCR-ABL, ALL, where it's the classical model of oncogene addiction. And so I'm sure you all know this better than I do because, as Paul brought up, I am a chemical physicist pretending to be a cancer biologist, so please don't call me out on that. You get this fusion between ABL1 and BCR or breakpoint cluster region. This leads to a block in B cell differentiation, and in adults, it comes with a poor prognosis. Now, what's so cool about where we are in sort of the world of chemistry at this point is that actually we have tons of different inhibitors, including third-generation inhibitors with non-overlapping sites that can go after sort of this kinase domain that's part of this fusion. We even have drugs that can go after other binding sites, like this myristat binding site, like ABL01, which give you the idea that maybe we could use combinations to get total suppression of this oncogene as a way of making it impossible to evolve resistance. And so could you, the question becomes, use something like ponatinib in combination with ABL01 as a way of basically killing all of your tumors by making it so you could not evolve resistance, sort of that synthetic dual hit lethality. And so this is work that Pete, Andrew, and Michelle did in combination with Mark Markami over the DFCI, where we started out taking some PDX models and we made sure that they recapitulated what you would see in patients. So we put them on ponatinib. You can see that in each case, the burden of cancer goes down, but all of them eventually relapse. And so since ponatinib as an individual agent doesn't work, you could ask yourself, well, what happens if we do this myristat binding site ABL01, or we do ponatinib, or we do the combination, or we do vehicle? And surprisingly, what we find when we do this is that the combination doesn't do much better. ABL01 does a little better than vehicle. Ponatinib drives, a pretty good increase in survival, but eventually all the mice end up dying. Same thing when we add the pair. And so you might ask, if we're hitting these things and we can't evolve resistance through BCR-ABL, what is happening here? How are we getting inhibition? So if we go and look pre and post-treatment in our mice, what we find is that there are a few patterns of mutation that emerge. So in some, we see a movement where we go from having lower mutations in STAT5 to activation of STAT5 pathway. In others, we see accumulation of mutations in ERK. And actually, interestingly, we see a third group in which there's no accumulation of mutations whatsoever, none that we can detect looking across the entire genome. And so we have these three patterns that are showing up, which is STAT, RAS, and none whatsoever, even after whole genome sequencing or whole exome sequencing. Now, what's so interesting for people that know B-cell biology much better than I do, because I've been learning a lot of this, is that these two pathways couple into the state of cells. So it turns out that STAT5 signaling is really important during the pro-B cell stage of B cell differentiation. This is a more immature-like state, whereas RAS and ERK signaling is really important for survival when you're at this more differentiated state in this pre-B cell-like state. So if we go and we look at these cells by immunophenotyping, we find that there's actually an accumulation of STAT5 mutants in the cells that are less developmentally mature, whereas those that have ERK or no mutations actually turn out to be more developmentally mature. So you're probably looking at this and you're saying to yourself, well, what you showed me is pretreatment, and then you showed me progression. But what's really important is understanding what happens at minimal residual disease. This tells us about how cells actually evolve resistance. It tells us something about what is happening that enables you to get to progression. And so what we decided to do is to take one of each of these mechanisms, an ABL mutant, a RAS mutant, and a no mutant, and look at what was happening at MRD by profiling those cells using SmartSeq. So we could do both transcriptional phenotyping and genotyping. So I'm just going to tell you some quick vignettes because of the amount of time that we have. But in our first model, what we found is that our cells at pretreatment started off being, relatively more immature. As we move to remission at MRD and the progression, we can see that they become more differentiated. In this model, we can see that there's no accumulation of KRAS or NRAS mutants or any things in that pathway when we compare pretreatment to MRD to progression. So what we're seeing is the way in which this particular model seems to get around the idea of BCR-ABL inhibition using a third-generation TKI is through differentiating to a more mature cell. And so this is strange because in all the things that you would think about in the literature, it's usually moving back to a more stem-like state that actually enables you to evolve resistance. But here, at least it seems like it's the opposite. So what about our other models? Well, if we go look at this model right here, we can see that we were already pretty developmentally mature. We stayed there throughout the various course of progression. This is a place where, we have few mutants early, we get 1 or 2 at MRD, and then they proliferate and grow out at progression. And this is because, remember, the RAS pathway is a place where, or RAS is very active in these more developmentally mature-like cells. So this is a place where we think we have a compatibility between cell state and mutation. If we look at the final model, what we see is that it started off being much more progenitor-like. As we progressed, at remission, it moves a little bit more mature, and then finally at progression, it's a little more mature than it was before. And when we look at the mutations that exist, we can see that there's actually, a good amount of KRAS and NRAS mutants, and at MRD, there's a huge enrichment of them. But funnily enough, we actually get a dramatic decrease in the amount of KRAS and NRAS mutants that we have when we get to progression. And so what we think is happening here is that actually RAS is active in these more developmentally immature states, which we can see when we go and dive and look. So we have a lot of progenitor-like cells, and we think that actually the RAS pathway isn't very beneficial in this particular state. And to get at this a little bit more, we can go and look and see what's active in the cells that have RAS mutants at MRD. And what we see are a number of genes that many of you in the audience probably recognize here that are telling us that these cells are actually activating senescence programs. And so what this does is it gives us a place where we can think about our cells at MRD as sort of falling on a more progenitor-like, more senescent state, or being more previously-like, so more developmentally mature, and being less senescent. So if this is how we get there, you might ask, well, could we target states? We talked about the idea of targeting states in classical and basal. Could we do the same thing here? Well, what we can do is we can look at the gene programs that are active in these cells, that are ways in which these cells are exploring to try and get around drugged. In all of them, we see this ubiquitous immune response program, which I'm not showing here, but we see differential activation of a stress autophagy program and a pre-BCR signaling program. And what that does is it lets us think about our cells on sort of the senescence axis right here. And when we look at where these programs overlay, it's really stress autophagy showing up at the high senescence and pre-BCR signaling showing up in these more poised, low senescent cells. So if you go look at a wiring diagram, what you can figure out is that actually there are drugs where we could go after pre-BCR signaling at a proximal site. So using fostamatinib, for example, to go after SYK. And if we wanted to get further down, we could hit stress response using something like losmapimod to go after P38. And so the natural hypothesis that comes out of this is we just have to understand the developmental state of our cells, and then we can do a better job driving deep remission. So we can go and test this using our PDX mice. We can say, well, let's try the combination therapy where we take a mouse and we either use ponatinib plus ABL-001 as our control, or one of these two drugs hitting a state-specific response. And so if we look at our first model right here, we see we have upregulation of stress. And so you think that losmapimod plus ponatinib is going to work better than the other combinations. And I wouldn't be talking about it here unless it does. It's that combination that drives much deeper remission when we look at involvement in bone marrow. So now for truth in advertising, does this work when we look at a model that has exactly the same or different state? So here we see pre-BCR signaling is up. So that tells us we should be using fostamatinib in combination with ponatinib as opposed to the other two. And once again, it works in exactly the way you would expect. And so I think that what I wanted to tell you out of this is that the interesting thing about progression on these third generation TKIs is that, it can happen via mutational or transcriptional responses, that it looks like there's some co-opting of these these developmental programs that we have to think about, and that mutational fitness depends upon the state you're in, because once again, it depends upon whether you're actively using that pathway. And so if we think about what state we're in, we can think about a better way of targeting that vulnerability to drive deeper remission. So, these were two little vignettes, but you're probably asking yourself, well, how do we think about moving into a much larger regime where we, think more broadly about what are mechanisms that exist? So if I think about what I told you very briefly about in PDAC, I would have thought about it as a vertical stack right here, where we're first looking at in vivo reference maps, and then we're building models that accurately recapitulate what we see, and then testing drugs to see whether or not they actually work. Could you do this for other cancers? Well, Andrew and Mifra, in combination with the NCI, have been doing this for a number of the different TCGA samples, so there'll be data that'll be up and available soon from multiple different tumors showing us reference states that we could use for these. Interestingly, even though these tumors are very different, we're seeing lots of convergent states, like that same basal cell state that I told you about in PDAC, showing up in multiple different cancers, suggesting that there might be some common targetable biology. Now, if we want to get to looking at models, how do we think through these intrinsic and extrinsic factors? It's hard, but there are ways of going after it. So on the intrinsic side, we can take advantage of some of the advances in cellular manipulation. So for example, we can take ORFs and we can overexpress things that we think might drive one state or another. So P63 overexpression always drives our models to be more basal-like. GATA6 drives us to be more classical-like. And if we want to understand the full landscape, we don't have to do it one at a time. We can actually take advantage of barcoded TF-ORF libraries, like Evelyn and Walla have been doing in combination with Julia Tuong from Bang Zhang's group, where by putting these in and then sorting cells based upon their phenotype, we can figure out which ORFs are actually driving specific states. Now, this is nice because we have a barcode, so all we have to do is sort and sequence barcodes. How do you do this when we're thinking about extrinsic things like cytokines, chemokines, or drugs? Well, this gets hard because there are lots of things you want to test, but this is where Connor and Ben came up with a crazy idea that just kind of works. So what we do is we imagine that we want to take 8 different things. So imagine these are cytokines in the environment, anything that you want. If we wanted to screen them conventionally, we would have to do, say, 4 replicates. This means 32 samples. What we began to wonder is, did we have to do that? Could we instead pool together our different drugs as a way of reducing the amount of samples that we needed? And the idea here is simple. If we make sure that we don't see the same pairs very often, we can treat this like a linear deconvolution problem, where we make measurements, we know our design matrix, and then we solve for what the impact is of each compound. This may sound insane, but it works. So we can take those same PDAC organoids, we can dump on 68 of those cytokines that I was showing you in that paracrine signaling plot in pools of 5, do a couple references, so our TGF-beta and our gamma, see what it does to gene expression, and then deconvolve the impact of each. If we run this in 2 replicates in one plate and another, you see that we get our TGF-beta and our interferon gamma coming up again. We also see a number of things that you might have expected, like TNF-alpha, IL-1 beta, IL-1 alpha. We also see really weird things like IL-4 and 13, so type 2 immune cytokines that we never would have thought about in the context of PDAC. So before I tell you a story about this, I'll just tell you that we can validate this. So if we go back and we test this in single kit, we get exactly the response we would have expected from the majority of them, including IL-4 and 13. Now, I think that one of the most important things that I learned from the study was not just that we could compress these things, but that the responses were very different than we might have imagined. So if we go look at the IL-4/13 drugs or genes that come out of our PDAC models, it's 133 genes that keep on turning on every time we do this. But if I look at the signature databases that exist, like Reactome or BioCarta or anything that would show up in a paper, I've got 5 genes or 0 genes or 1 gene in terms of an overlap. And so, this is not surprising when you think about it. Different epigenetics, different proteins, different responses. But it turns out that it leads us to wildly different interpretations. So here we see that actually stratification by IL-4/13 leads to difference in outcomes. So high IL-4, IL-13 activation in our PDAC cells is actually associated with worse outcome, whereas we would have never picked this up in other contexts. You might say, well, this is just a weird one. And I'll say, no, if I go back and I look at things that you would have expected to work better, like TNF or TGF-beta or interferon gamma, it's the same story in each context. And so what this really means is that when we're mining genomics data, trying to do hypergeometric enrichment, so you have all your genes, and then you try and figure out what is the enrichment, what is the signature, in many places we're probably missing what we need because we don't have the right references. And we have to do a better job of creating that. So getting to the final piece of this, how do we think about going after state-specific dependencies? Well, you can imagine you've seen compression now with drugs, particularly ones that are not always bioactive. We can do crazy compression. With a repurposed drug library, we've done 80 drugs at once, so we can figure out what the major effects are. When not all are bioactive, we can actually do even higher. And we can add this in combination with things that I was showing you before, like adding gamma to figure out in what cases do you get better survival and in what cases do you actually get better killing. Now, you might say, well, why do I think we should keep on doing this? Why do I think state-specific responses are going to be so important? It's because if we go back and we mine CRISPR dependency data, what we can find is that if we ignore the tumor of origin and instead focus on these RNA expression states, so are you basal or are you more mesenchymal-like, we could have predicted what were going to be the liabilities to, CRISPR in a dish. And so I think there's going to be a lot of room for us to think about the idea of targeting states, but only if we understand some of the things that came up today. What controls those states? How did they change when you move to different microenvironments? And how can we go after them? So a lot of what we've been thinking about is how do we complete the square, sort of creating these pan-ecosystem, our cancer ecosystem models that help us to understand what's driving specific states in cancers, creating models that represent them, and then using those as ways of testing drugs and testing the plasticity of the models to evade those drugs. And I think that where we want to get to at the end of the day is what I like to call, I call it, well, I'm not going to have a good term for it on the spot. We'll just call it targeting tumor tissues as opposed to DNA mutations. And I think what I mean by that is we can imagine being in a world where we do things like activate T cells to secrete gamma, which controls plasticity in cancer cells, so we can now come in with our MAP kinase inhibitors to drive better outcomes. So, really going after eradication of the tumor as opposed to a specific genetic lesion. And so with that, I'll thank my team because they actually do all the science. I don't do any of it anymore. But most importantly, I want to thank patients and families because it's not lost on me that the only reason that we can do these experiments is because patients and their families partner with us at a time when they're incredibly vulnerable. And I think we owe a tremendous amount of respect to those individuals and to make sure that what we do actually benefits them, their families, and their wishes. This is not my work. This is a huge collaboration with Scott, Chris, Doug, Bill, Andy, Brian, and David. And I'll also thank our funders who've been incredibly generous and hopefully will continue to be. And thank you all for coming today. It's great to see you all. I'll take any questions. Good.
Super. So we've got time for a couple questions. Yes.
Thank you. Great talk. So targeting self-death is a very exciting idea. So my question is that can we use partial reprogramming strategy to target tumors or microenvironment cells to re-educate tumors or redifferentiation or transdifferentiation tumors back into normal state is a possible solution for cancer treatment in the future?
So I think it's a super interesting thing. So I interpret your question two ways, and tell me if I misinterpret this. So I think in some ways we can totally target differentiation states of tumors. People have done this in differentiation therapies for a long time. So that's one thing that we can imagine. The other thing that we can imagine is that, yes, we can try and rationally reprogram the tumor microenvironment to slow progression, to make cells more targetable. I think the big thing that we have to learn, which is complicated, is what is the hierarchy of responses. So, I didn't dive into this in PDAC, but we see associations with specific myeloid states. We see associations with T cells. And the question becomes, does the tumor cell program the myeloid cell or does the myeloid cell program the tumor? And what we need to understand is not just the plasticity and the timescale for it, as came up in some of the nice talks earlier today, but also to understand, what are the timescales that are associated with multiple different populations and what is their potential to reprogram a specific cell, and how will that play out as you move to different sites? The lung is very different than the liver. We see predictable changes in state when we look at metastases to the different sites. So I think we have to learn these rules that govern how tissue ecosystems are wired, but it's a phenomenal time to start thinking about this and to leverage a lot of the things that have been presented already in some of these technologies to gain a more holistic understanding of how tumor ecosystems work.
Thank you.
Please.
Alex, really nice talk. You highlighted a really interesting point that the gene signatures that you observe and what's already there in the existing databases, there's a mismatch in that. Where do you think cell types come into this role? Like, different cell populations have different, signatures when it comes to these pathways, or?
So I think that, when we think about what really defines a cell type, and I'll give a short answer to this, which I'm not good at, and then I'll happily ramble for a very long time afterwards. I think of cell types as being defined by the genes they express, which is defined by the epigenetics. And so you've got a combination of accessible DNA, maybe a couple different mutations in proteins. And so when you come in with a stimulus, you're going to have different molecules at different levels helping to execute some of those signaling cascades, which is going to result in different responses. And so I think that we're always going to see different signatures. And there's really cool computational opportunities in thinking about how to use transfer learning to map between them and to use existing large ATAC-seq databases or existing bulk proteomic data sets to predict those. But I think it's a missed opportunity, a place where we could actually really accelerate our understanding of all of the things that we're measuring now by just fixing our reference data sets.
Thank you.
Hi, great talk. So I have a question. Biopsies taken from different sites of PDAC models, whether you start working with them ex vivo, do you have any indications of whether hypoxia is happening at different levels in them and whether it might affect on the response to treatments, more resistant, less resistant?
So I don't have a great answer on the hypoxia piece because in most of the things that we're doing, we're growing them as organoids or cell lines, and we didn't play a lot with oxygen. We played with multiple different ways of growing them. If we grow them big enough, they end up dying. So I've seen a lot of things in the data that we've looked at where changes in nutrient availability change state. And it turns out that we see more movement to this co-stressor-like state when there's metabolic stress. So my gut response would be, my guess is that under reduced oxygen, you're going to see a transition to more of this co-expressing state. And some of the stuff I know about myeloid cells and the way in which they couple in would make me believe that's true as well. But I need to test it. I don't know.
I see. Thank you.
Let's do one more, and then we'll close.
Great talk. You've proved once again that no one does precision medicine. And I'm not only joking, that's the truth. So when you do your screens with targeted or untargeted drugs, do you analyze how much output you've inhibited as a function of concentration?
So in some places, it's a great question. So in some places, we've started to look at multiple different concentrations to figure out what we need to get to. But getting to the idea of the fact that we need to screen multiple molecules, multiple cytokines, multiple models makes it really hard. And so
it's also very key. So Gideon Bolak did the best and only experiment on this in people. You need about 90% of ERK inhibition for a RAF inhibitor to work in melanoma.
Yeah.
Not 89.
Was a great talk. So I was particularly interested in the different tumor dependency upon TGF-beta. So when you switch from classical to basal like PDAC cancer. And so I was wondering whether you have investigated further why when you switch to the basal state, they become more dependent on the MAP kinase inhibitor.
So we haven't yet. We have some follow-up experiments where we're actually working our way through multiple different nodes in the signaling cascade using ORFs and using CRISPR-Cas9 to knock things down and upregulate them in the opposite order. So we're really trying to get at that mechanism to understand what's underlying it and whether or not there are multiple pathways that one would need to drug because that's where you'd want to go next.
Thank you.
Thanks.
All right, well, let's thank all our speakers from this morning.